Welcome to the Everything Epigenetics podcast, where we discuss DNA regulation and the insights it can tell you about your health. I'm Hannah Went, and I'm the founder of Everything Epigenetics. Today, my guest is Kristen McGreevy. She just had a paper published recently in Aging, which will be our main discussion today. She created a DNA methylation fit age. So we're able to predict different physical fitness outcomes and measurements from looking at our DNA methylation and epigenetic regulation. A little introduction to Kristen. She studied biology and biostatistics at UNC Chapel Hill, where she graduated as a first-generation student in the Honors College. While there, she researched how water bears survive extremes that the materials they are made of cannot even withstand. And after that, she traveled out to UCLA and attended school there to receive her MS in biostatistics. And she is currently finishing her PhD and expected to graduate in the fall of this year. Her research focuses on the epigenetics of aging and building biomarkers to measure aging driven by lifestyle factors. If you are interested in physical fitness at all, learning how our methylation is being affected by it, this is one that you will want to tune in and listen to. Now for my guest, Kristen McGreevy. Welcome to the Everything Epigenetics podcast, Kristen. I'm so excited to have you. Thank you for being on today.
Thank you for having me. I'm excited to be here as well.
Yeah, and I always like to start off by, learning a little bit about you. Of course, I have my little introduction that I gave, but for those listening, can you just tell us a little bit about your journey? How did you become interested in biostatistics and epigenetics? You're currently getting your PhD. So even maybe for people who are listening and thinking about going into this space, what type of advice would you give them? All of the above.
Yeah, so I think my trajectory into where I am is relatively linear. I was fortunate to find what I liked in high school. So my one friend told me that they could increase their odds of winning a raffle by folding their ticket. And I thought, wait a minute, I need to know this because the pizza raffle's coming up. So I did a stats experiment in high school to figure that out. And that was kind of when I realized that biostatistics or statistics is the best tool to answer our research questions. And then I had more exposure to that in undergrad when I worked in a wet lab. And I realized that being at a bench all day long is a little bit difficult, especially when you're going to be focusing on one protein at one cell membrane for years of your life. And so I found biostatistics as the best avenue for me to understand research and to explore what's happening with the data. And then when I was going into grad school, I knew I loved epigenetics then. So I was teaching genetics in undergrad and I loved epigenetics. I thought it was the new version of genetics, really, where it's the modification of our genome and we can change things based on our environment, our habits. And so I found Steve Horvath, and I found his research and I thought, wow, this is amazing. I'm going to go to California. I'm going to do my grad studies at UCLA and hopefully work with him. And so I came out here. That's ultimately the story that arose. And that's why I'm staying. I started working with Steve for my master's and that went well. So then I decided to stay for my PhD.
I love that. You knew what you wanted. You went after it. You got it. So you're right. That's such a linear path, but such a great story. And where are you from? Where did you move from to go to California?
Yeah, so I grew up in Pittsburgh and then I did my undergrad in North Carolina. So ultimately, I moved from either one of those areas really out here.
Yeah. Okay. Gotcha. And do you remember when was the first time you were taught epigenetics in school? Do you ever remember? Like I think back and I think maybe it was like one sentence in my textbook in undergrad and that was it.
Right. I think it was kind of similar where it was in my genetics class. It was towards the end of the quarter and they brought up this idea of epigenetics and DNA methylation. And I was just blown away by it. So immediately I went to the textbook and I started doing all of my research that I could because I thought it was just the most interesting thing that we could modify how the genome was actually going to be read. And Yeah, I agree. It wasn't extremely well taught, so you had to go to find these other resources.
Yeah, I was just curious. I think yeah, people have a similar history with epigenetics in terms of just like their normal routine schooling that they attended. And interesting that you liked biostatistics from the beginning as well, because I always say I just have a biology undergrad degree, but if I could go back and do it all again, I would have definitely gone into like computer science or computer training in the biostatistics. And because those wet labs become very repetitive, or sitting at a bench all day, which is what I did. So yeah, jumping right into it, you have an amazing paper that was just published. This is perfect timing for our interview, but this paper is titled DNA Methylation Fit Age. So it's a biological age indicator incorporating physical fitness. So that is really what we're going to dive into. But I first want to start just with the discussion of physical fitness and relating that to, epigenetics. So, how do you define physical fitness in your paper, and why is this important to epigenetics? And you can relate it back to aging, of course, too.
Right. Yeah. Well, thank you for saying it. It's a great paper. I appreciate that a lot. So I think there's typically 5 classical areas of physical fitness. One is this body composition. Another is flexibility. Then we also have strength. We have muscle endurance, and then cardiorespiratory endurance. And I think most well known within those tends to be strength and endurance, because we think about strength training and we think about endurance training. I think that physical fitness is a combination of all of those. And so you need to have a little bit of all of these different measures of fitness in order to be very fit and physically fit. And so flexibility, I think, is pretty, general. So it's how much movement you can have. And we know that this movement decreases as you get older. So range of motion and mobility. Body composition is more so about how you have fat or muscle or bone distributed across your body. And commonly we think of that being measured through BMI. But what's very informative is where that fat is distributed. And so abdominal fat, and seeing that changes as we age. And that exercise itself is very important for this, in that if you exercise, you can actually decrease the abdominal fat specifically and have better body composition in relationship to these aging phenotypes. And then muscular endurance is really, ability to like contract a muscle, pretty simple, repeatedly. And then cardiorespiratory fitness is your lungs, your heart, all of that working together in order to provide oxygen to your body. And then I think strength is pretty straightforward. It's, this force. It's, we see that lack of exercise is a significant risk factor for a lot of different things, and that we see that strength is extremely important for, long-term longevity. And so physical fitness generally, as you know, declines with aging, and that people who preserve fitness as they age are at a lower risk of a lot of different diseases and tend to live longer and healthier lives. And then we see that these changes in fitness seem to relate to these molecular processes. So you mentioned that at the end, how it starts to be related to the epigenome. So we see these differences in athletes versus controls in what the DNA methylation patterns are. And we also see that there seems to be this long-term effect that is accumulated in the epigenome from a lifelong time of exercise.
Sure. And, what I do when in my daily job is work with a lot of healthcare providers and, trying to talk to them about recommendations for having a healthy epigenome, you could say. And we always bring up physical fitness, but that one's a little hard to talk about because I would say the research is still limited behind it. That's why I love your paper so much. So you just named those 5 areas of physical fitness. Would you say one is more important than the other, or would you prioritize one over the other?
Well, I think if I was going to choose one, it's going to be a relatively biased answer because I've been a runner my entire life. And so I think that's part of my influence. But I also am basing my answer based on this research. So a lot of research has looked more at endurance and strength. And I see that strength training, especially for women, is extremely beneficial. So I would say having women do more weight training, that would increase their longevity and health substantially. And then endurance is also very much looked at, and that seems to have in my opinion, a stronger effect on these biological changes. So both of those, having good endurance and good strength, I think are, very high priority. But there's also a lot of research that looks at more of this mobility and flexibility as being this good indicator of how you're going to respond to this interventions later in life, or that's very much related to cognitive decline, having your neurons firing at a frequency. So there's a lot of different parts to which one I might prefer, but I think overall, I would say strength and endurance are my top picks.
Gotcha. My mind went to the strength, of course. I'm a weightlifter and getting more into running, which is exciting because I used to run a lot and play a lot of sports, but I definitely need to work on more of like the flexibility and mobility.
I think that's all of us runners.
Yeah, definitely like the hot yoga. You're like, oh, why am I doing this or wasting my time? But it's so important as well. So I think just, you're right, a nice healthy balance. And there's research really with the endurance and strength, but more to come on the flexibility and mobility. So we'll see. So I always love hearing how papers like this come to fruition. I love hearing the background on them, what questions were being asked in the lab when you were first creating the paper. And in your paper, you even mentioned there are 3 lines of evidence that support a focus on DNA methylation to develop biomarkers of fitness and these age-related changes in fitness. So I want to elaborate on that. What prompted to build, these DNA methylation estimates of fitness parameters?
Yeah, so I think it first started from us observing that many of our DNA methylation biomarkers that we were using didn't seem to perform as well as expected in athletes or people who had long-term exercise regimes. So we were a little surprised to see that we weren't seeing such a strong signal in these biomarkers because these people we know to be healthier. And so then we started thinking, well, why is this happening? And other researchers have demonstrated this as well, that we don't really see this benefit or difference in athletes versus other people until about the age of 60 or older. But we can physically see all of these differences at a younger age point. So we started thinking, well, ultimately, what can we build in order to distinguish this difference and, notice it at an earlier time point? And so we first said, okay, we know that DNA methylation changes as we age. We've had so many different biomarkers built from that. And so we've had these high precision algorithms, especially for aging, which are also the epigenetic clocks. So that was part one. We know DNA methylation is changing, that would be good for some type of aging biomarker. And then we also know that epigenetic clocks have been improved by incorporating physiological data. So, for example, we have phenoage, germage, etc., that incorporate these smoking pack years, blood cell counts, plasma proteins into the prediction algorithms. So incorporating some other aspect of somebody's health is good for predicting these age-related conditions. And so that would suggest that physical fitness would be a very natural incorporated into these DNA methylation biomarkers, but none of the current biomarkers included them. And then the last part of this is, well, we also know that there's this emerging evidence that these epigenetic clocks are sensitive to the lifestyle factors. So you can improve your epigenome. And we also see that differences in your fitness parameters are reflected in the DNA methylation data. And so we start to have this growing body of evidence that blood DNA methylation carries information related to physical fitness. So we wanted to see, could we actually build some type of biomarker of fitness through blood DNA methylation, which, really we don't even know. We didn't think that there would be a strong signal because it's blood.
Oh, really?
Yeah, well, because fitness you would think might be more related to any of your other organs, such as, muscle or even adipose. But we are able to see a signal in blood, which was surprising. It's not an enormous signal. It's not like our correlations are 1, but we still do pick up a signal and it relates to a bunch of different outcomes.
Oh, yeah. Interesting. Yeah, but you're right. Once you really think about it, you may not hypothesize that you would see a signal in blood and maybe different, muscle tissue or something of the sort. So yeah, that was a good point.
Thank you.
And you just mentioned you use blood, DNA methylation biomarkers.
Mm-hmm.
So hopefully everyone listening knows why blood is so important to use when looking at these epigenetic clocks. I've addressed that with people in my previous interviews. So we're going to skip over that point. But you look at these biomarkers of 4 fitness parameters. So you have your gait speed, which is your walking speed, your hand grip strength, your forced expiratory volume in 1 second, and maximal oxygen uptake or VO2 max, so a measure of cardiorespiratory fitness. That's correct, right? Those 4 markers?
Yes, exactly.
Perfect. And this is something, I've been very curious about. So why did you choose those 4 DNA methylation fitness biomarkers? And then, you can go into them a little bit in depth, maybe tell our listeners a little bit about what each of those mean.
Sure. So when we were choosing these, we needed to balance 3 different factors. One was measuring physical fitness across the different areas of fitness. So we talked about all of those 5 different main areas. And then the other part is, well, can we choose these fitness parameters that are well researched and that we know to be associated with aging and age-related outcomes? So narrowing down what measurements we can take of fitness that we also know are relating to aging. And then the last part that we had to factor in is, well, we need to build a biomarker of fitness measures that have not been developed yet. So we need to provide something that's new to the field. And so we balanced all of those. And we also were, limited by what we had available in the data sets, but we had a lot of data sets. So thankfully we weren't super limited. And so we ended up choosing those 4 biomarkers, like you mentioned. So gait speed, grip strength, forced expiratory volume 1, FEV1, and VO2 max. So gait speed, walking speed, it's really a measure of this mobility. And we see that it's a combination of flexibility and this lower limb strength. So it's really capturing 2 different parts of our fitness, this flexibility aspect and then this strength aspect. Hand grip strength is very well researched, and we see that if you have muscle weakness or weak grip strength, you have accelerated biological age. It's also related very much to just maximal bench press, is just a cool side part about physical fitness. And then we also see that it's related to premature aging, mortality, disability, etc. So we knew grip strength was going to be important, and that's very much just a measure of that muscular strength. FEV1 is a measure of lung function, amount of air that you can force from your lungs in 1 second. And people say that it's more so a measure of this plasticity in your diaphragm and that it's related to your core stability. And it measures really how much air you can move in and out of your lungs at a time. And so that's going to very much relate to how much oxygen you can get throughout your body when you're exercising. VO2 max, I think, is my favorite measure. So it's your maximal oxygen consumption. It's really a measure of your cardiovascular or cardiorespiratory health and your aerobic endurance. So it's the amount of oxygen or the volume of oxygen that your body processes with incremental exercise. And ultimately, this is considered one of the best indicators of an athlete's physical capacity, and it's the international standard for physical capacity. So we see that VO2 max is really, quite important, and we see this routinely related to aging phenotypes that this will decline over time, but people who exercise seem to preserve VO2 max and have these better outcomes.
Yeah, why is that one your favorite? I have to know.
I think ultimately it's harder to measure, but I think that it gives you a very good idea of your intrinsic fitness from this measure of, you're taking into account your heart, but you also have to be able to do this exercise in order to take the VO2 max measurement, because you're going into harder and harder exercise as you're doing it. And people who are much older, sometimes they can't take this VO2 max measurement because it's not safe, because you're going into harder exercise regimes. And so you get to really see this transition of how you're starting from the beginning, how you're going up. So you can also relate this to, well, how am I feeling if you have a heart rate that's lower in the beginning, but then after a couple of minutes, you're like, wow, I'm super fatigued. And so VO2 max is really, I think, a good measurement to say, well, can you even do this type of exercise? Do you have the strength? Do you have the flexibility, the mobility? And then how well can you do it for how long?
Gotcha. Isn't it right? You put on like a mask and you're on a treadmill. Is that correct? Yeah. Okay.
Yeah, so
I know
that's a very common way.
okay.
Yeah.
And there are other ways maybe to measure it too?
Yeah, so there's another way. I mean, it's very similar to that. You can also be on a bike. And then there's also another one, which is called the chest or step test, where you're really like going up and down stairs. And then if you don't have any way to do physical tests itself, then there is some type of conversion between your heart rate and, these other variables. But that's not as good of a signal because that's very much dependent on a lot of different factors. It's not the best measure of VO2 max. The best ones are exactly what you're saying, wearing the mask, running, or wearing the mask biking.
Yeah, I've never measured mine, so I'm super curious. Now I'm thinking, am I even able, would I be able to do it? Would I be able to even measure it? So yeah, I'm going to get that tested at some point. Were there any fitness parameters that you really wanted to include, but you didn't have enough data on them or didn't make the cut? I know you said you were lucky with what you had, but was there something that you're like, oh, it was just so close?
Yeah, I think that's, a great question because ultimately I built a lot of biomarkers in this process. So some of the biomarkers that I built, I just didn't have variables in the validation data sets. So I wasn't able to see, are these actually measuring what I want? So for instance, I also built a biomarker for the maximum distance that you could jump in height. And so that, yeah, so that would be a great marker, right? Because it's, it's strength, it's, maybe a little bit of flexibility, maybe a little bit of endurance since you're taking that measurement so many times. And ultimately, there was not very good variables to correlate that to check in the validation data set. So, I mean, gait speed, sure, might be kind of related to jump max, but at the end of the day, that wasn't a strong enough relationship to say whether or not this was going to be informative. And then the other biomarkers that I've considered was a measure of body composition. So I didn't talk about body composition in any of these DNA methylation fitness biomarkers. So I considered making a one for BMI or this adiposity around the waist. Didn't have adiposity around the waist, so couldn't look at that. And then there's also methylation biomarkers for BMI already in using blood. And so ultimately, I was thinking it might be better to build one using adipose DNA methylation instead.
Gotcha. Yeah. Very cool. That jump max is super interesting. It brings me back to my track days in high school when I would do like the long jump, and the long jump's not, I wasn't very good at it by any means, but it was really fun just, seeing how far you could jump. So that's super interesting. And to expand even further, what makes these biomarkers different from other DNA methylation biomarkers?
Yeah, so I think the different DNA methylation biomarkers are incorporating different information or are measuring something different. So the Dunedin pace of aging, that's very much a measurement of how your aging is changing. So your biological age and how that's, pace. Pheno age is another one that it's commonly looked at, which incorporates this phenotypic information, but it's all types of phenotypic information in the blood. So you have proteins and blood cell information like albumin, glucose, lymphocyte percentages, etc. And then Grim age is a measurement also built in blood, but it's measuring and incorporating these different plasma proteins and pack years. So a measurement of how much people have been smoking. And so ultimately, all of these different biomarkers haven't incorporated this physical fitness component to them yet. They are incorporating different parts of the blood or different parts of this phenotypic, relationship to somebody's aging and their biological age, but they haven't incorporated the physical fitness. And I think that's ultimately what makes these ones and, the later part of this paper, Fit Age, more novel and distinct.
Awesome. Yeah, no, that's great. It's super unique insights that you're looking at here. So you're taking, you now have these DNA methylation biomarkers to predict your gait speed, your grip strength, the FEV1, and the VO2 max. So you have kind of these segregated clocks, right? You could call them epigenetic clocks for this fitness. But you're using then these biomarkers to develop this very novel DNA methylation fitness related biological age indicator. So, the title of your paper, the DNA Methylation Fit Age, and this is really quantifying the relationship between physical fitness and biological aging processes. So can you explain this? What's this DNA methylation fit age?
Yeah, absolutely. And what you said is exactly right. So we first are introducing these DNA methylation fitness biomarkers, and then we're combining them to get an estimate of somebody's biological age from this perception of physical fitness. So we are incorporating 3 of our fitness biomarkers. We include gait speed, grip max, and VO2 max. We excluded FEV1 because when we looked at the different models in the end, it didn't really seem to improve things as much. So we just took the parsimonious model. And so we're incorporating those 3 fitness parameters along with grim age, the mortality risk estimate, and we're combining those through the method Clamero-Duval, and all of this together comes up with a biological age estimate. So this fit age is really a biological age incorporating strength, mobility, and cardiorespiratory fitness.
Awesome. Just taking some notes here as we're chatting. It's super interesting. Yeah. And I want to talk about the population you use. So what population or cohorts did you use to actually, I don't know if they were different when you created, the 4 clocks and then the DNA methylation fit age, or can you talk about that?
Yeah, sure. So we used 3 data sets in the training part of this. So the Framingham Heart Study, FHS, and Baltimore Longitudinal Study of Aging, BLSA. And then we also had a novel study, which is Budapest, which are my co-first author, Zolt Radok, had collected all of this data in Budapest on these long-term rowing athletes and controls. So these people are all performed in this rowing regatta at some point in their life. So they're like very, very good rowing athletes. And then there's like these controls who were age and sex matched. So we used those 3 different data sets to combine them into a training set. So the Framingham Heart Study is, a study of your heart. It's a very well-studied population. A lot of different biomarkers have been developed using this, and it's a large data set, so over 1,000 people in it. Baltimore Longitudinal Study of Aging, we have multiple time points on people, so ultimately we're just using that first measurement so that we're not getting an increase of the correlation. But all of these people are generally healthy in the FHS and the BLSA, and then we have that novel fitness component in the athlete part from the Budapest. So those were all our training data sets. And then our validation data sets ranged as well. So we looked in calorie, we looked in Women's Health Initiative. We have in Canty. We looked at a lot of different ones, including the Lothian birth cohorts. So overall, we had, I believe, 6 validation data sets that we were looking at how these different phenotypes related to our fitness biomarkers and ultimately this estimate of biological age.
Sure. That Budapest cohort you mentioned is super interesting. Are there a lot of cohorts of athletes like that, or is that hard and atypical to find?
It's very hard to find. It's quite sad. But ultimately, most of the studies, people aren't going to be these extreme athletes. So I think typically these clinical trials or these studies are trying to enroll healthier people that aren't like super healthy or too far from the norm. And so it was actually very difficult to collect this data and to get something like that, even if you go onto Geo to look for these different databases, it was hard to find athletes that also had DNA methylation taken, because typically when people are looking at athletes, they're more inclined to say, well, what's the recovery period? Or how fast are they going to be? How much longer do they have to be in this sport? They're more so focused on these other variables, not necessarily biological age, because they already know they're fit. They already know that they're healthy. They don't really need these type of measurements. So it was hard to get. And interestingly, next year, we're going to have my collaborators are going to have a data set that they are fortunate and very kind to let me share, where they are sampling 80 gold Olympic medalists and then age and sex matched controls. And so they're looking at DNA methylation, and that will be very exciting.
Okay, yeah, that's super interesting. We're going to have to chat more about that offline. I'm super interested in hearing a little bit more of that. But like you said, there's, not really much we know or can look at as it relates to DNA methylation. So I think that will just be a huge value to everyone doing research in the space and knowing those specific outcomes. So that sounds extremely exciting. Mm-hmm. And leading off, what's the application of DNA methylation fit age? I don't know if it's better to ask, how is that actually related to athletes or physical activity and different age related phenotypes?
Yeah, so I think that there's two main things that fit age has been shown in the paper, at least, to relate to. One is these age-related phenotypes, and then physical fitness in the expected directions. So what we first did is built this age-adjusted version of fit age. So we're calling that fit age acceleration. And as you know, it's pretty common to just talk about age acceleration values. So really how deviant you are from your expected age. And so positive values tend to be bad because that means you'd be older than expected, and negative values are good. And so we use this age-adjusted version, and then we look at how that relates to a variety of things. And we see that it is predictive of mortality, coronary heart disease, and we also see it's related to earlier age at menopause, more comorbidities. All of those were if you had a larger fit age or a higher age acceleration, you had an increased risk for that. And so if I'll just give you one example, mortality, we had a hazard ratio of 1.07. And so that means that if you have a 10-year acceleration in fit age, that corresponds to an almost doubling of your mortality risk compared to someone of the same age and sex. And so that just shows that fit age seems to be related to some of these age-related conditions. And then the opposite was also true, where if you had a better fit age or a younger age acceleration, then you had better outcomes. So you were associated with disease-free status and lower cholesterol. And so all of that is really showing that, okay, we see that fit age is related to these aging outcomes. And it seems that if you are fitter, then you have better age-related phenotypes. And then we also saw for the fitness part of view that if you were more physically fit, then you had a younger fit age. And this was true in both men and women. So we saw that if you looked at people with just intermediate amount of exercise compared to people who had none, we were able to see that just a small amount of physical activity really was improving your fit age and then also those individual fitness biomarkers. It wasn't a huge increase. We see like a 0.33 year decrease. So your age acceleration, 0.33 years decrease with just a change from 0 to low to intermediate physical activity levels. But then we also looked in a sample of bodybuilders. So we were able to find Polish collaborators who had sampled bodybuilders and DNA methylation. And we saw that bodybuilders were biologically younger and more physically fit. So their fit age value on average was 2.7 years younger in the male bodybuilders compared to controls. And so that's great. And then we also saw that DNA methylation VO2 max was 0.4 milliliters per kilogram per second better in these bodybuilders as well. So we are able to see this relationship to physical fitness in these athletes, so the bodybuilders, but then also in just regular people in our validation data sets. So our validation data sets didn't have athletes in them, but we saw that if people had just a slight increase in their physical activity level, then you had better outcomes.
Oh, wow. That's very promising. It makes you, want to go work out. I already worked out this morning, but makes me feel guilty now for skipping some days. So even just, a little bit of movement is really great. Am I correct in maybe making that conclusion?
Yeah, and I think that we are starting to have more research that shows just even marginal, very slight increases in your physical activity really have large effects. Where I don't remember what the study was, but I remember that within it, they only had a 1 day a week increase in physical activity. So they said, just do 1 more day a week of physical activity. And they were able to see in people who were starting at zero exercise, an improvement of their biomarkers. So I think that ultimately what we're starting to see is that just even a moderate amount of exercise is super beneficial for your health.
Yeah, I think that's super hopeful as well, right? Because it's you get in this routine and maybe you fall off the routine, and it's really hard to get back into the routine once you're off, right? I know, at least personally, I feel that way. So it just pushes that point further that just a little bit goes a long way. Recently, a couple changes that I made is I felt just like I was sitting all day and I wanted to move a little bit more. I only took like a 15-minute walk from my when I'm working 9 to 5. And, that just seems ridiculous. So I took the initiative, got a stand-up desk, and then got one of those walking pads to go underneath of it. And I thought it'd be a lot harder than it actually seems. And it's great. I'm getting in, a ton more steps. I can't stand sitting down all day. Now I just want to be standing up. I remember on the way to work hearing something on the radio that said like sitting is the new smoking. And that's what really started looking into it. Yeah. So I think we'll see a big shift in trend toward, more of that healthy living, hopefully. Right. Because at least in especially here in the States, we're very stressed out. We're very, sedentary. We're not getting in a lot of movement. So just that hopeful piece to hold on to, that just a little bit actually goes a long way, I think is a great silver lining.
Yeah, I completely agree. And just another thing that I think is interesting, I'm going to share through that I've learned through this research, is that you mentioned that it's hard for you to get back into exercise afterwards, when you have this, period off. And I think that's true for a lot of people. But then what we also are able to see, even at the epigenetic level, is that people who are, exercise or athletes for a long period of time, if you have this short amount of time, maybe a few months that you're not exercising, it's a lot easier for you to get back into it and get to your max or where you were before, rather than somebody who would just be starting at what your level was at that time period where you're resuming exercise. And so this curve of how long it takes you to get back is really shortened and abbreviated for people who were athletes or exercising previously. And they say that this is from this epigenetic memory. And so there seems to be a signal that's restored and retained in your DNA methylation or other epigenetic components that translate to you getting back to your peak exercise performance quicker after a period of rest.
That's very interesting. Yeah, because like mentally it's hard for me to get back up and get in the gym. But actually once I'm in the gym after I've taken like a couple day break or even a week or so, I don't know if I'm like on vacation and not in my normal routine, I do almost feel like a little bit stronger and like I didn't lose a lot of that muscle memory. I actually do, because I think also our bodies need breaks, right? We can't be in go mode all the time. So that's interesting. Yeah, I find that to be true for me, at least.
Yeah, and I found it to be true for me as well. I had a concussion last year, and I was out for 2 months of exercising, and I, would exercise, 4 to 5 days a week. And I noticed that once I got back into it, very quick, and I had the same experience. I was like, wow, muscle memory. I was able to get back here so quickly. And then I had a conversation with our collaborators, and they started to share this with me. So then I started doing a little bit of research into it. And thankfully, the research backed my experience, which was I was able to get back into my peak performance quickly, even after taking 2 months off.
Yeah, it's a real thing. I like it. Epigenetic muscle memory. So I'll keep that in mind next time, again, you feel guilty for like taking a break or taking days off. Kristen, is there anything else you want to add? Anything that these biomarkers contribute to the field of aging and epigenetics? I know they contribute a lot we've already talked about, but anything else?
Yeah, I mean, I think really I see them as providing a physical fitness component to our epigenetic clocks and being able to show that our blood epigenome changes do accompany physical fitness. And so I think that this is really what they're providing. It's giving us a molecular understanding of the exercise benefits, and that hopefully these will allow us to see more of a difference in athletic populations at an earlier age, instead of having to wait for around age 60 to see these benefits of exercise really kicking in. So hopefully we'll be able to see these as tools to either measure interventions in, exercise-based regimes, or then also be able to distinguish athletes versus other people at earlier time points to actually show that there is some type of difference biologically.
Sure. And how can we actually trust that the DNA methylation fit age is a biomarker of aging?
Yeah, I think it comes down to how you view what aging is. So I think if you, people have different theories of aging, and ultimately fit age is built on two main requirements. So in order to build this, I mentioned the Clemmer-Duval method. And so in order to use this method, you first have to say, well, we believe that biological age is centered on chronological age. So that means that if we sampled everybody who is chronologically aged 60 in the population, the average of their biological age would be 60. And so if we just believe that biological age is centered on chronological age and that we have deviations from that, that's one part. And then the other part is that our biomarkers that we're measuring, so these different fitness biomarkers, are actually functions of biological age, that they're changing with biological age, and that biological age is actually the predictor and can be used to estimate the biological age or the biomarker itself. And so those are two of the requirements when we're building our models. And so if you believe that those two things are true, then ultimately you can start to believe that the methods that we use to build fit age are valid. And so one is just saying, okay, I trust these methods and I accept these requirements of biological age to build this. But then the next part, I think the trust comes down to, well, do you believe that things are related to aging if we see a relationship to age-related phenotypes, which is everything that I've been able to observe so far. So, we looked at mortality, we looked at type 2 diabetes, menopause, cancer, cholesterol, disease-free status, etc. And we see that all of these are related in the expected direction. But we haven't been able to explore other aspects of aging. So, we didn't look specifically at these different inflammation markers or, senescence. So if you have a different theory of aging, then maybe you wouldn't believe it or something. But at this point, it just hasn't been validated in those steps. So I think, in summary, I think it just comes down to, do you believe in the assumptions or requirements when we built it? And do you think that aging can be well summarized with the relationship to age-related phenotypes?
Yeah, I think that's just an important question, right? One we should ask, all of the researchers who are creating these papers and different insights into aging, because like you said, it varies from person to person and kind of their methodologies that they use. And aging is just so complex. So we're learning more as people like yourself and other researchers are diving into these experiments. Mm-hmm. And we're almost getting to the end there, but a couple more questions. I think I say this all the time, epigenetics is extremely exciting, but I think it's equally frustrating because it's so new. There's a lot that we don't know. So what do we not know about these biomarkers? What wouldn't they tell us?
Yeah, so right now we haven't been able to evaluate them in a much younger population. So most of the time when we think of athletes, we think of people who are, maybe, like 18 to like 25, a small range. We think of them as being pretty young people, and we haven't been able to evaluate them in a super young population. So one is, well, we need to go to younger people and see, is this something that validates at a younger age frame? Because we're able to see it in these lifelong athletes or people who have been athletes for a long period of time. And so we have, in that regard, more time to modify your epigenome. So we need to see how it performs in a younger population. But then we also haven't seen how it performs based on fitness interventions. So if somebody goes into an exercise regime, can we see in 3 or 6 months, maybe even a year, a change in these biomarkers? We don't know yet. And so ultimately, I would say those are the two biggest limitations as well. How does it perform in younger people? And do we know if this is going to be modified based on exercise?
Sure. Great points. And I'm sure we'll find out, the answers to those questions again, as we build from this type of again, amazing work that you've done. So you have this paper, it's finally published. What are you focusing on now? What other research are you conducting? Are you adding on top of this, switching gears?
Mm-hmm. Yeah, so I am in a few different projects. One, as I mentioned earlier, is looking at these gold Olympic medalists and controls. So I think that will be quite interesting to see how the epigenome is changing there. There's other projects that also might be quite interesting to your listeners. So I'm looking at DNA methylation in muscle and adipose tissue in the calorie study, which was the clinical trial that people restricted their caloric intake by 25% for 2 years. And so I'm looking at not just how DNA methylation is changing, but also how your baseline DNA methylation is predictive of your responsiveness to the treatment. So, we have caloric restriction, and that if you restrict your calories, you will lose weight. That's just, the science behind it. But then you stop losing weight at some point because you adjust your BMI. And so seeing what that responsiveness is, we're hoping is going to be predictive of what your baseline DNA methylation is, and then seeing, well, if you can improve that based on some other lifestyle trait. So I think that's the other component that's quite interesting. And then I'm still finishing my dissertation, and so I'm also trying to improve imputation of DNA methylation by using a transformation procedure, which hopefully I will be able to share with all researchers, similar to how all of these biomarkers are available too.
Awesome. Yeah, thanks for that, Kristen. Sounds like you are doing a lot of different projects that are super exciting. I'm going to have you back on here a little bit more about that once we were able to. This has been great. I really appreciate you being on and giving us your expertise and knowledge in this field. One question I ask everyone at the end, it's a curveball question. Kristen, if you could be any animal in the world, what would you be and why?
Ooh, okay. Well,
just a little fun one.
I think anytime that we try to say what animal we want to be at the top of the food chain somewhere, because we're afraid of other animals trying to eat us. And so, a super simple, probably predictable answer would be a hawk or something, because you want to be at the top of the food chain. And also, I think everybody really wants to fly. So that I used to think I wanted to be a dolphin, but I realized that being in the ocean is actually kind of scary. So not a dolphin anymore.
There you go. Well, it seems like you've thought about the question before, and then you have reasoning too to be at the top of the food chain. So I love it. We've come to the end of this amazing podcast interview. For listeners who want to connect, where can they find you? How can researchers access this information, or these algorithms if they want to look at other biomarkers or insights with their data?
Yeah, so you can connect with me on LinkedIn. I'm Kristen McGreevy. You search me and that'll come up. I'm also, on Instagram, Twitter, etc. So whatever avenue people really want to contact me, they can. Email, also fine. kristenmay@ucla.edu. And then to get all of these different biomarkers, I have all of the code and functions to use them on GitHub. So if you go to GitHub, github.com/Kristen McGreevy/DNAM Fit Age, you'll find all of the code and programs in order to make these biomarkers yourself, as long as you have DNA methylation data. And then the paper, as you mentioned, is published in Aging Albany. So if people want to read the manuscript themselves, they can go there.
Perfect. And I'll make sure I'll put all of that in the show notes so people know where to find you and they're able to access your great work. So thank you again, Kristen. I really appreciate your time. Thanks for everyone listening and joining the Everything Epigenetics podcast. Remember, you have control over your DNA regulation and epigenetics. So tune in next time to learn more. Thanks so much, Kristen.
Yeah, thank you, Hannah. Thank you so much for having me.